Okay, so hello everyone. I'm really happy to have the opportunity to share my work with you today, and we will dive together into my ongoing intracranial investigation of the heartbeat evoked response across vigilance states. So the aim of this project is to investigate the neural network underlying the autonomic control of the cardiac function, and to do that, I'm using the heartbeat evoked response to probe the brain in wakefulness and sleep because they are believed to reflect the cardiac afferent processing. So if you look at scalp EEG studies who look at heartbeat evoked response during wakefulness, they found that they were associated with a widespread cortical network, but not a lot of studies have tried to look at this neural source during sleep, heartbeat evoked response during sleep, and almost exclusively with scalp EEG. However, we know that scalp EEG has two key limitations: a limited spatial precision, so it's harder to find the neural source of this heartbeat response, and also artifact contamination, mainly by the cardiac field artifact and the pulse artifact. So to overcome this limitation, I'm using intracranial data from SEEG. So SEEG is an invasive technique used with people with drug-resistant epilepsy to try to pinpoint where originate precisely the seizure on the brain. So we put really thin electrodes, sometimes really deep inside the brain, and it allows us to explore the brain with a good temporal and spatial precision and less artifact contamination. So with this method, Perrin and al. Look at neural sources for heartbeat response during wakefulness, and they found that it was associated with a broad network also, with the insular cortex as one of the main sources for this heartbeat response. And if you take together other results from cardiac manifestation during seizure, for example, you can see this network with some subcortical relays like the amygdala and the hippocampus, and other cortical parts of the brain who are implicated in the cardiac autonomic regulation. So my work is in line with this work, and I will try to look at this neural source during both wakefulness and sleep. To do that, I'm using data from 10 epileptic patients, so it represents 200 points of contact in regions of interest. And I'm comparing response during wakefulness, but also during N2 sleep, N3 sleep, and REM sleep. I highlight here two key important steps in the preprocessing to get rid of the artifact. So the bipolar montage to reference my electrode is supposed to remove all the cardiac field artifact, and the pulse artifact is supposedly associated with 2 Hz frequency, and so I apply a 4 Hz high-pass filter to get rid of this artifact. I'm averaging the signal time lock to the air peak of the signal to create the heartbeat evoked response, and then to validate the responsive contact, I will compare my original signal to a distribution of a signal not time lock, a random average. So I will shift every onset individually for every trial and create an average random, and then I will create 1,000 average random to create a distribution to compare my signal with. So now we can start this exploratory approach and start to ask a question. So the first one for me was, which brain region is really involved in this network and in the processing of the heartbeat? And so I will show you an overview of all the responsive contacts that I have obtained during by vigilance states and by brain regions. So you have here the list of all the brain regions that I have explored so far in a bottom-up organization. And keep in mind that I don't have the same amount of electrodes in all the regions because every exploration intracranial is unique. But anyways, I found some responsive contact in different layers of the brain, so in subcortical relay like the amygdala and the hippocampus, but also in more cortical part like the frontal lobe. And the insular cortex appears to be the main source in my results for this heartbeat evoked response. The second result that I want to highlight here is we found heartbeat evoked response in all the different vigilance states that I have analyzed so far. And if we look closely, we can have some hints about my second question. How does the activity of this network might vary across vigilance states? Because we can see there is some region with a similar number of responsive contact through vigilance states, like the insular cortex or the temporal lobe, but we have also other parts of the brain who react only during wakefulness or only during sleep. So to investigate this, we can zoom into one region because it seems to be an effect region specific. So for the sake of the presentation, I will just go into the insular cortex. Here you can see an overview over time of all the responsive activations that I have obtained through the 70 electrodes in the insular cortex. So I don't know how it works to point, but anyways, if you look at one time point, you can see the number of responsive contact I have obtained in N3 state, N2 state, and wakefulness. So basically here you have this tendency to have more activation and longer activation with increasing sleep depth. So to verify this potential modulation, we can zoom into examples of heartbeat evoked response that I have obtained. So here is an average of an heartbeat evoked response in one channel, in one contact point in the posterior insular cortex, first across all vigilance states, and then if we look at every different vigilance states, we can see that we have a similar pattern of activation, and you can see that when the line is thicker, it means that it appears to be significant in my surrogate distribution analysis. And we have here two patterns significant, one between 100 milliseconds and 200 milliseconds, and another one between 300 and 400. Here we can have maybe the impression that there is no modulation by the vigilance states in posterior insular cortex because we have really a similar pattern in terms of timing and amplitude. But it's a bit more complex because if we look at another channel still in the posterior insular cortex from another patient, we have here a signal who appears to be really similar until the air peak, and then who seems to elicit a response modulate by the states of vigilance because we have really a bigger amplitude with increasing sleep depth. So it's a bit hard to conclude at this state. As I mentioned, it's an ongoing investigation, so I have a few hypotheses. It can be maybe a spatial difference between the electrode and individual differences, maybe some artifacts that I didn't notice until now. So I don't know. But to conclude, I found some heartbeat evoked response across all the vigilance states in various brain regions, so at subcortical level and also at cortical level. The preliminary results suggest that might be a modulation by the vigilance states and maybe in a region-specific manner, but it's too soon to conclude to something. So I need more data to probe the entire brain and to have a better representation of all the parts of the brain. And maybe in the future, conduct connectivity analysis to underlay to discover the dynamics inside this network. So thank you all for your attention, and a special thanks to my supervisor and colleagues. Thank you.
Thank you. Do we have any questions? Yeah, sure. Go right ahead.
Yeah, thank you. Very nice data. I just would like to bring attention to one very important point we yesterday briefly discussed with Marcia. So there is a kind of belief that if you go inside the brain, then the ECG heart field artifact is not as strong, for instance. But that actually should be another way around, because if you think about lead field matrix on the scalp, yeah, it includes also skull, which has a very high impedance. But here, that's a little bit different. So you will have stronger signal, yes, because you're closer to source, but you should have also enormously strong heart field-related artifact. Actually, it should be even stronger. So therefore, I would like you just a little bit to comment on why do you think that bipolar derivation should get rid of it? Because bipolar is a spatial filter. It depends how your gradient is oriented. You might pick it up or you might enhance it. So what's the rationale behind it? And have you tried to look at your evoked responses looking the monopolar, basically, derivations? Thank you.
Okay, so yeah, I mean it's a fundamental limit for all heartbeat evoked potential. We try to find ways, standardized method to get rid of this artifact, but at some point we can't be sure. The theory in intracranial data is when you apply a bipolar montage, you are supposedly suppressing all the noise coming from other parts than a 3 millimeter radius around your new electrodes. So I try to follow knowledge that we know and theory that we install, but yes, at some point I can't be sure that I get rid of all these artifacts, and I try to find ways to control more, but for now we don't know this way.
We can talk later more.
Yeah.
So there's a question back.
Could you hear me? Yeah. I saw your patients cohort, and I have a question about that, because when I doing some patient study, I can see some baseline difference. So, did you see any, or did you browse any inter-subject difference at the baseline, or this baseline may take some effects in the final results?
I didn't check for now, because the first limit with intracranial studies is the number of patients. It's really hard to obtain data, and usually studies with intracranial patients, it's less than 10 patients. Often it's 1, 2, 3 patients. So my first objective is to construct a big data set of patients, because it's really rare in intracranial studies. And then I will start to look at inter-subject differences that might explain my result. But yeah, it's not my priority right now. I'm sure there is some individual differences, but the first aim is to have a representative data set.
Because especially for the epilepsy data, I saw there's a quite difference
Yeah.
cross subjects.
Yeah, I think.
Thank you.